So, in last few classes, we are discussing about microbes, right? We have completed the discussion about viruses. We have completed the discussion about bacteria, and even we have completed the discussion about archaeas. All those things that we have already discussed, and even the videos are made available in our YouTube channel. Okay. For any doubts, you can check the YouTube videos, and if there is any doubt persists, you can ask here. Okay. So in today's class, we are discussing about bacterial growth curve, which is one of the important topic. Bacterial growth curve. Okay, it's about the bacterial growth curve that we are going to discuss in today's class. Okay, listen. So, if I am looking for the growth curve of bacteria, if we are looking for the growth curve of bacteria with respect to the time, how the number of cells are increasing in bacteria with respect to the time, the number of viable cells. What is the number of viable cells? If you are making a graph with the log number of viable cells, or simply the number of viable cells. The number of viable cells means that is living cells with respect to the time. You will be getting a graph in this manner. And the graph will be in this manner. Initially, the graph will be moving in this manner. And after that, you can see a growth in this manner. And after that, you can see a growth in this manner. And after the growth will decline. What is happening here, the first phase of growth is known as lag phase. The first phase of growth is known as lag phase. What is happening during this phase, during lag phase? What is happening during lag phase? During lag phase, a bacteria, so a number of bacteria are there in a medium. We are looking, a number of bacteria are there in, suppose small numbers of bacteria are there. We are looking for their growth in this particular vessel. So initially, the growth will be very slow. That phase is known as lag phase. What is occurring during the lag phase? There isn't that much division during this phase, right? There isn't that much reproduction. During this phase, preparation or synthesis of molecules are occurring. During lag phase, what happens? You can see the preparation. You can see the preparation or synthesis of molecules occurs. Synthesis of molecules occurs during this phase. So first phase, that is known as the lag phase. During lag phase, the bacteria will be producing. What? They will be preparing molecules which is responsible for their growth. So we can check for the second phase. What is happening during the second phase? This phase is known as, after lag phase, this growth phase is known as log or exponential phase. This phase is known as log, or it is also known as exponential. Log or exponential phase. This phase is known as log phase or exponential phase. Or it is also known as tropo phase. It is enough. Lag or exponential phase. It is known as lag. This is the lag phase, and this is known as log or exponential phase. What is happening during the lag phase? During lag phase, the preparation was happening, right? So during log phase, what happens? During log phase, there is maximum growth, right? So during the log phase, there will be growth of the bacteria, or bacteria will divide faster, or even you can tell the exponential growth of bacteria. Exponential growth, you can tell it as a exponential growth. During the log phase, you can see the exponential growth by using the nutrients. Nutrients are already synthesized, right? So by using the nutrients, you can see exponential growth during the log phase. That is the second phase. It is known as log phase. So what is happening after log phase or exponential phase? The next phase is known as stationary phase. The next phase is known as stationary. This phase is known as stationary, or it is also known as idiophase. Stationary or idiophase. What is happening during the stationary or idiophase? We can look what is happening during the stationary phase. Check what is happening during the stationary phase. During the stationary phase, there will be the depletion of nutrients. So the growth of bacteria is becoming dormant. There isn't any further growth because of the depletion of nutrients occurs. All the nutrients will be utilized by depletion of nutrients. All the nutrients are utilized by the bacteria for their growth. So now what happened, there is no nutrient. That's why the growth is declined in this phase. What is happening during the last phase? This phase is known as death phase. This phase is known as death phase or declined phase. Death or declined. You can call it as death phase or declined phase. It is a last phase in the growth of bacteria. And what is happening during the death phase or declined phase? During the death phase. During the death phase, no doubt at all. During the death phase, there will be the completely nutrients completely over. Nutrients are completely finished. Nutrients are completely over. So what will happen? The bacteria will die. That is occurring during this phase. This is the main 4 stages of bacteria. Or during the bacterial growth, you can see lag phase, log phase, exponential phase, and also death phases. During lag phase, the bacteria will synthesize the nutrients which is required for the division of cells. During the log or exponential phase, the bacteria will multiply faster and they will produce all the nutrients required for, during the log phase, during the log phase, when the bacteria are growing, during the log phase, when the bacteria are growing, what will happen, during log, whenever the bacteria are multiplying, what they will do, they produce during log, primary metabolites are produced. During log phase, you can see primary metabolites are produced. During log phase, you can see primary metabolites are produced for what, produced for their survival. For the bacteria to survive, they requires primary metabolites. What is the difference between primary metabolite and secondary metabolite? Primary metabolite means that metabolite will be having known role in that organism's body. Secondary metabolites, in the sense, secondary metabolites will not be having a role in that organism's body, but it will be beneficial for other organisms. So, for example, if you are looking into a rubber plant, from rubber, rubber plant, rubber is coming, right? Rubber, that is watery fluid is a fluid is coming with this whitish in color. That is, we are solidifying that into rubber, right? For producing tires and all. If you are looking for that plant, because of that rubber, it is not at all useful for that plant. Such chemical substances are known as secondary metabolites. Secondary metabolites are coming from plants and microbes, but it will not be useful for that organism. Still, we don't know if there is any role for that particular chemical substance. For example, certain plants will produce pigments, carotenoids. Certain plants will produce gums. Certain plants will produce polymeric substances, for example, cellulose. Certain plants will produce scents, smelling substances, perfumes. All these things are coming from plants, right? But we don't know whether the plant is having any whether the plant is or the microbe is having any benefit out of that particular chemical, that we don't know. Such chemicals are known as secondary metabolites. Whereas primary metabolite means which is very much required for their normal growth and development or reproduction. Such chemicals are produced during the log phase or the exponential phase while the bacteria is growing. For their survival, they will produce primary metabolite. If you are looking for primary metabolite, protein is an example for primary metabolite, or carbohydrate is an example for primary metabolite. Such chemicals are known as primary metabolite, that is produced during the log or exponential phase. The secondary metabolites, when a plant is produced or when a microbe is producing secondary metabolite, that is most probably during the stationary phase. During stationary phase, they will produce secondary metabolite, secondary metabolites. The secondary metabolites will be produced during which phase, during stationary phase only. So certain chemicals in the case of microbes, if you are looking, that may be for their defense. It may be for their defense, this secondary metabolite may be for their defense. Anyhow, this is about the stages, and during these stages, they will produce all these substances. Okay. We have already told that the primary metabolites are produced during the exponential phase, right? But their synthesis will be started. Their synthesis will be started during the log phase. Their synthesis will be started. They are produced during the log phase, but their production starts in lag phase. So if you're looking for primary metabolite, the primary metabolites are produced in log phase, but their synthesis starts. In the case of primary metabolite, their synthesis starts in the lag phase. During lag phase, their synthesis will be starting. Okay. By using this, if I am asking a question, okay, if I am simply asking a thing, during the growth of a bacteria in a particular medium, when will be the what I can ask, when will be the penicillin, when a microorganism will produce penicillin? When a microorganism will produce penicillin, we know very well who is producing a penicillin. That is produced by a bacteria that is known as Staphylococcus. The Staphylococcus is producing the penicillin to defeat against a particular fungi, right? That Penicillium notatum. It is it is produced by the Penicillium notatum against Staphylococcus. Produced by the Penicillium notatum against Staphylococcus. So the penicillin will be produced during which phase? Whether it will be produced during lag or log phase? Tell me. The penicillin will be produced during lag, log, or stationary?
Stationary.
What is penicillin? Penicillin is actually an antibiotic, right? Antibiotic. It can kill microorganisms. So the penicillin, which is coming from the Penicillium notatum, blue mold, which is killing Staphylococcus, we know very well. So the penicillin is a secondary metabolite. So penicillin will be produced during the stationary phase. If you are looking for organic acids, or if you are looking for ethanol, such substances, which is required for their growth and metabolism, such chemicals will be produced during such chemicals are known as primary metabolite. Their production starts in lag phase and their synthesis will continue during the exponential phase also. This is the bacterial growth curve. Whether it is clear?
Yes, sir.
Okay.
Yes, sir.
Okay. So if we are moving further, look, I'm representing another graph here. I'm showing another graph, okay? One more graph I am showing here. This is another representation of this chemical substance coming from the body of this type of organisms, of microorganism. Okay. First of all, I am showing the biomass here. That, as we have already told, this is the biomass. This is the first phase. Then there is a next phase. Then there is a next phase. Okay. What is this represent? This is representing the biomass. This is representing the biomass or the amount of that organism or the number of that organism. You can see lag here, you can see log here, and you can see the stationary phase here. And after that, there will be one decline phase also. Okay. If you are looking for penicillin, okay, if you are looking for penicillin along with the graph, along with the biomass, if you are looking for penicillin, okay, if you are looking for penicillin, you will be getting a graph in this manner. During the initial time, the growth of penicillin will be minimal. But, during with this phase, during this phase, the growth of penicillin will be increases and the growth of penicillin will be very high during the stationary phase. So this is the case of secondary metabolite. So for example, this is penicillin, okay? The case of penicillin will be in this manner. Its growth will be increasing in this manner. Okay. We know very well it is the lag phase. This one is a lag phase and this one is a log phase and this one is a stationary phase. This is a stationary phase. All these things we know very well. Okay, clear. So if you're looking for the lactose, okay, lactose, what will happen for lactose? Lactose initially, lactose will be very high in the medium. Lactose is a food, okay, suppose it is a food. The amount of lactose will be very high during the initial time. And what will happen with the growth of the bacteria? With the growth of the bacteria, the amount of penicillin will be, the amount of penicillin that will be declining. The amount of penicillin will be declining, isn't it? So it will happen. So here you can see the growth of bacteria, where you can see the different type of chemical substances. What happens during the lag phase? And if you are looking for another chemical, you can look for the case of ammonia also. So the NH3, the case of NH3 will be also high during the lag phase, and by course of time, the amount of NH3 will also decline. The amount of ammonia, that will also declines. This is the graph, a simple graph by using the previous thing itself. Okay. So into the media, okay. Suppose into the media, if I am adding, when this was happening, okay, this was happening, till this stage it was happening, okay, till this thing it was happening, okay. So this thing happening means lag phase is there and log phase was happening. If more bacteria, bacteria if more bacteria are put into, if I am adding more bacteria are put into same media, if I am putting the more amount of bacteria into the same media, what will happen, then there will not be the preparatory stage, because during lag phase only they were preparing for growth, right? They were preparing molecules for the growth, right? So as it is already prepared and as it is already available in the media, if you are adding more bacteria into the media, what will happen, no lag phase will be there. No lag phase occurs. So no lag phase will be occurs if I am adding more amount of bacteria into the same media. Whether it is clear?
Yes, sir.
Okay. Ready. So during the first division, okay, with the first division, what will happen with the first division, okay? So initially I was having a single bacteria. When the single bacteria is getting dividing, the single bacteria is getting dividing, I will get two bacteria. This is our first division. During second division, what I will get during the second division, I am having two bacteria there, right? So I will get four bacteria, isn't it? I will get four bacteria. During third division, if I am looking for the third division, I am having four bacteria with me. So during third division, I will be getting eight bacteria. During next division, that is during the fourth division, the eight bacteria will be producing 16 bacteria. During the next division, that is during the fifth division, the 16 bacteria will be producing 32 bacteria. In this manner, this will continue, isn't it? Ready. So if you are looking, the 2 is 2 raised to 1, right? 2 is 2 raised to 1. And 4 is 2 raised to 2, right? 4 is 2 raised to 2. 8 is 2 raised to 3. 8 is 2 raised to 3. 16 is 2 raised to 4. And 32 is 2 raised to 5, isn't it? In this manner, only the division continues. So we can write in this manner also. At the 0th division, if there is no division, 0th division, there will be one cell, isn't it? That can be represented as 2 raised to 0. So 2 raised to 0, 2 raised to 1, 2 raised to 2, 2 raised to 3, 2 raised to 4, 2 raised to 5. In this manner only, the division of the bacteria proceeds, isn't it? Okay. So if the initial cell is 1, if the initial cell is 1, so at the beginning, if in the media, if there is only one bacteria, okay, if the initial cell is 1, the number of cells, the number of bacterial cells, the number of cells after nth generation, after nth generation, the number of cells will be 2 raised to n. Whether it is clear, the number of bacteria will be 2 raised to n. Okay, for example, in 16th generation, in 16th generation, what will be the number of bacteria? The answer will be 2 raised to 16. that will be the number of bacteria in 16th generation. Okay. In 0th generation, 2 raised to 0. In first generation, 2 raised to 1. In second generation, 2 raised to 2. Third generation, 2 raised to 3. Fourth generation, 2 raised to 4. Fifth generation, 2 raised to 5. In this manner, in nth generation, if you are looking for the nth generation, the number of cells will be 2 raised to n. So in 16th generation, it will be 2 raised to 16. Whether it is clear?
Yes, sir.
Okay. So by using that, okay, we are going to make an equation here. So NT, what is NT? You know, NT is the NT, that is the number of cells at time t. When the time is t, you know, the number of bacterial cell will be equal to N0 into 2 raised to n. So what is N0? You know, N0 is the initial cell. N0 will be initial cell. N0 is the initial cells. And n is equal to the number of generation. N is the number of generation. So you are getting an equation here. Nt is equal to N0 into 2 raised to n, where N0 is the initial number of cells and Nt is the result, that is the number of cells at time t, and n is the number of generation. Whether it is clear? Okay. So if we are looking I'm telling a question here, okay. N0 is equal to 10 raised to 3. N0 is equal to 10 raised to 3 cells per ml. And the lag time is equal to lag time. Lag time is also provided. Lag time, that is 10 minutes. 10 minutes. 10 minutes is a lag time. What is lag time? During lag time, they won't divide, right? After the lag time, they will divide. So after 10 minutes, they will divide. That is lag time is the generation time. That is clear, right? That is a generation time. It is a generation time. So during lag time, they won't divide. We know very well. After lag time, they will divide. That means with the 10 minutes, they will divide. Okay. So it is a generation time. That is the time required. What is generation time? That is a time taken by a cell to divide. Time taken by a cell to divide. That is a lag time. That is 10 minutes. So the question is N0 already given. 0 is already given. And the question towards us, what is the question, you know, that is what will be the cell density. What is the cell density? What is the cell density? That is a number of cell at 30 minutes. What will be the cell density at 30 minutes? That is a question. What is the cell density at 30 minutes? Okay. Okay, come on. Try to do. So N0 is given to you. What will be the cell density at 30 minutes? That is a question. What will be the cell density at? Okay, anyhow, I will do this question. Okay. If I am asking, what will be the value of n here? What will be the value of n here? N is a number of generation, right? Whether it is 1, 2, or 3. What will be the n here? The value of n, 1 or 2 or 3. Come on. N is a number of generations. Hello? Hello, am I audible?
Sir, 3, sir.
Who is telling 3?
Darshini, sir.
Okay, Darshini. How you got the 3 as the answer?
Sir, if 30 minutes total time na, அது வந்து 10 minutes வந்துட்டு the total generation time na, so 1 to 10 ல வந்துட்டு ஒரு இது நடக்கும், so 10 to 20 இன்னொன்னு நடக்கும், 20 to 30, if 30 is considerable, 3 generations, sir.
If I am telling that is wrong,
Okay, sir.
Darshini.
Yes, sir.
If I am telling that is wrong, what you will tell?
Okay, sir.
Come on.
அது தப்புனா 2, sir.
Why?
Whyனா அதேதான் 1ல இருந்து 10 வரைக்கும் ஒரு இது, ஸோ 10ல இருந்து 20 வரைக்கும் இது, ஸோ 20 to 30 இஸ் அது 30 minutes அங்க முடியறதுனால if we can't consider that 2.
Darshini.
Yes, sir.
Darshini. Total time
Yes,
is
sir.
30 minutes, right?
Yes, sir.
First 10 minutes will be?
One.
What is the first 10 minutes?
The time taken for the cell to divide, the lag phase.
That is lag, right?
Yes, sir.
First 10 minutes is lag, right?
Yes, sir.
Whether the bacteria will divide during that stage?
No.
So, what will be N? What will happen during first 0 minute to 10 minute? What will happen? Whether it will
Nothing,
divide
sir.
or they will prepare their material.
They'll prepare
Prepare their
then
material.
the
Then 10
minute
to 20, what will happen?
10
One
to
generation.
20.
What will happen from 20 to 30?
They'll again divide.
Second generation. So,
Yes,
what
sir.
is the number of N here?
2.
2 is the N, isn't it?
Yes, sir.
What you are making stories for 30 minutes and all, during 30 minutes they won't complete. Okay.
Okay,
Listen
sir.
well. Okay. So now find out the answer. So if you are looking for the answer NT, we have to find out NT in the sense N30 that we are finding, right? N30 is equal to N0 into 2 raised to N. N0 into 2 raised to N. The value is already given. N0 is equal to 10 raised to 3. So 10 raised to 3 into 2 raised to 2. That is the answer. So we'll be getting the answer as 10 raised to 3 into 2 raised to 2. We can find it this manner. 2 raised to 2 is equal to 4. So 4 into 10 raised to 3 will be the answer. Whether it is clear?
Yes, sir. Yes, sir.
Okay. Everyone?
Sir, one doubt, sir.
Ask me.
Sir, next case க்கு போகாம next எப்படிங்க சார் டிவிஷனுக்கு போகும்?
Sorry?
சார் நெக்ஸ்ட் ஸ்டேஷனரி ஃபேஸ்க்கு தானங்க சார் போகணும். அது எப்படிங்க சார் திரும்பவும் டிவைட் ஆகும்?
If it is getting food, they will divide now.
Okay, sir.
If it is getting everything available in the medium, they will continue to divide. Whenever the sources decline, then only whenever the resources are limited, then only they will be going to the stationary and death phase. Okay. Otherwise, whether there will be any bacteria remains in the earth, all the bacteria from the beginning itself, they will enter into the stationary and they will die, right? There will not be any bacteria here. It's clear?
Thank you.
Okay. So we are proceeding further. Now listen. So look here, that is the NT is equal to N0 into 2 raised to N, right? N0 into 2 raised to N. If I am giving a log to both sides, okay, that is log NT is equal to log NT is equal to log N0 into log N0 into 2 raised to N. Log N0 into 2 raised to N. So in other means that is equal to, we have to multiply with the log, that is log N0 plus log N0 plus log 2 raised to N. So log, now log NT is equal to log N0 plus N log 2. N log 2. This N will come here. N log 2. So log NT minus, if I am doing log NT minus log N0, log NT minus log N0 will be equal to N log 2. It will be equal to N log 2. So I am looking for the number of generation. So if I have to get the number of generation, if I am having all the values, so the number of generation, what is the number of generation? If it is a question, number of generation, if it is a question, that is n is equal to, I can find easily log nt minus log n0 divided by log 2. By applying into this value, I can get the number of generation if they are provided with the nt is already given. What is the number of generation? For example, the number of generation at 30 minutes. The number of generation at 30 minutes, they are already given. For example, at 30 minutes is at n30. N30 is equal to 4 into 10 raised to 3. They are given like. And n0 at the 0th time, that is 10 raised to 3. They are given this also. Only this is enough. So you have to find out n30. They were in, so at 30 minutes, the number of generation, what is the generation number here? What is the n here? That you can easily find out by applying into this formula. The number of generation n will be equal to, we have to just apply, that is log 4 into 10 raised to 3 minus log 10 raised to 3 divided by log 2. Then you will be getting the value of n. N is the number of generation. So in order to produce 4 into 10 raised to 3 cells from 10 raised to 3, how many times the bacteria has undergone their division? That you can get from this equation. N is equal to log nt minus log n0 divided by log 2. Whether it is clear? Hello?
Sir, once more repeating, sir.
Okay. So, suppose a question in this manner. I am having a question. In a bacterial colony, there were a bacterial colony. A bacterial colony is there. In that bacterial colony, at initial time, there is 0 time at the beginning. When the bacterial colony, during the beginning time, there were 10 raised to 3 cells. 10 raised to 3 cells per ml was there. 10 raised to 3 cells were there at the beginning time. Okay. So this much amount of cells were there. After 30 minutes, 30 minutes are over. After 30 minutes, the number of cells are 4 into 10 raised to 3 cells are there. 4 into 10 raised to cells are there. 4 into 10 raised to 3 cells are there after 30 minutes. So the question is, in which generation that colony shows 4 into 10 raised to 3 cells? At the 0th generation, at the 0th time, there were 10 raised to 3 cells. From the 10 raised to 3 cells, from this 10 raised to 3 cells, the bacterial colony has to multiply into 4 into 10 raised to 3 cells, isn't it? So at the initial time, there were only 10 raised to 3 cells. Now it is after 30 minutes, they are having 4 into 10 raised to 3 cells are present. So our question is that the extent of division of this particular cells, whether it has undergone 1 division, or whether it has undergone 2 divisions, or whether it has undergone 3 division. What is the what I can say? What was the question? Okay, so the n is the question here. What is n? N is a number of generation. So what is the n in this question? If it is a question, okay. So after 1 division, what will be produced, and after 2 division, what will be produced, and after 3 division, what will be produced in this manner. From this, we can, as it is a previous question, we can easily tell it is a 3rd generation. After 3 division, they will produce 4 into 10 raised to 3 cells, that we can easily tell. But if the value is much more bigger, if it is like 14 into 10 raised to 3, what will be the division there? What is the generation? By which generation they will show 14 into 10 raised to 3 cells? If it is asking like that, we cannot easily find out the answer. So for that, we can make another equation. That equation that we are making from the previous one, the previous equation was nt is equal to n0 into 2 raised to n, right? Okay. So I am applying log into both the sides. That is log nt is equal to log nt is equal to log n0 log n0 into 2 raised to n. Log n0 into 2 raised to n. Okay. So if I am expanding that log this side, that is you can get log n0 plus log n0 plus log 2n. In this manner, you can expand this. Okay, again log nt is equal to log nt is equal to log n0, and you can expand this one in this manner. That is n into log 2. N into log 2. This is a mode of expansion of this. If I am subtracting the log n0 from log n0 from log nt, we will be getting as n log 2, n log 2. So from this, you can find out the number of generation n. N will be equal to log nt minus log n0 all divided by log 2, log 2, log 2. So if we are asked in this previous question, if we are asked the same thing, we can simply put here log into, for this question, the answer will be log 14 into 10 raised to 3 minus log 10 raised to 3 divided by log 10 raised to 3 divided by log 2. If you are doing that, if you are doing this thing, you will be getting the answer. But it will be very simple thing that we can easily apply. Such a type of questions will be asked there for your examination. So whether it is clear now, why we have find out n?
Yes, sir.
Okay. Everything is clear?
Yes, sir.
Okay. So if you are looking n0 is equal to n0 is equal to 35 cells. Okay, n0 is equal to 35 cells. Okay, nt is equal to 560 cells. 560 cells are the nt, and the doubling time or generation time, doubling time is equal to 60 minutes. 60 minutes is the doubling time. Okay. So in this question, we are asked about the n. Okay, if you are asked about the n, you can simply substitute log 560 minus log 35, isn't it? Log nt minus log n0, right? So log 560 minus log 35 divided by log 2. So you can find out in this manner. That will be log 560 minus log 35. You can write in this manner. Log 560 by log 35 divided by log 2 divided by log 2. That will be equal to, where is my calculator? $560 35. Anyone is having a mobile phone with you? Just 560 divided by 35. What is the value of 560 by 35? 35 will be coming here. In 56, that is 1.
16,
35. 16, right?
Yes,
so
sir.
that will be equal to log 16 divided by log 2 will be the answer. Log 16 divided by log 2, that is 4. Okay, that is 4. Okay, in option it will be shown like this. Okay, you will be having the option as log 16 divided by log 2. It will be the option. Okay, well, it is clear how to do that question?
Yes, sir.
Okay.
Yes, sir.
So we can find out from the same equation, we can make another equation also. That is a mean growth rate. What is a mean growth rate? How we can find the mean growth rate? Mean growth rate is the number of generation. Mean growth rate is a number of generation per unit time. Number of generation per unit time. What is the number of generation per unit time? That is mean growth rate. That is equal to mu. Mu is the n divided by t. Mu is n divided by t. So mu is n divided by t in the sense the value of n that we are already know. The value of n is log nt minus log n0 divided by log 2, isn't it? Log 2. So we have to just apply the t here. That is the mean growth rate. That is a number of generation per unit time. What is the number of generation per unit time that we are getting by using this equation? That is mu. The value of mu is n divided by t. The value of, or otherwise you can use this equation also. I hope this thing is clear.
Yes, sir.
Okay. So the response of microbes to their environment, we can just look for the microbial response to the environment. Microbial response to the environment. Few things that we have already discussed in previous class. Based on solute and water activity. Based on solute and water activity, we can just classify the microbes into osmotolerant are there, and halophilic organisms are there. Osmotolerant and halophilic organisms are there. Osmotolerance, they grows in a wide range of osmolarity. In a wide range of osmolarity, in a wide range of solute concentration, these organisms can grow. Osmotolerant. Halophilic organisms are salt-loving organisms. They are salt-loving. Halophilic organisms are salt-loving organisms that requires 2 molar NaCl. 2 molar NaCl is strictly required. 2 molar NaCl is strictly required for halophilic organisms. So you can classify them into osmotolerant organisms are there, halophilic organisms are there. Halophilic organisms, they will grow in a narrow range only, whereas osmotolerant organism, they can withstand a wide range of osmolarity. Okay, ready. So if you are looking for the growth rate with respect to the NaCl, okay, growth rate, if you are looking the growth rate, I am drawing another graph. Here I am representing the NaCl, value of NaCl is here, and the growth rate is here. Here it is a growth rate. Here it is a growth rate. Okay, growth rate is here and NaCl is here. So this will be the growth rate in the case of, this will be the growth. Oh, sorry, everything has gone. Okay, sorry. This will be the growth declining of non-halophilic. In non-halophilic organism, you can see a graph in this manner for non-halophilic organism. Non-halophilic organism means where there is high concentration of, if there is concentration of NaCl, then only they are. This is non-halophilic. Non-halophilic means they are not at all loving salt condition. Whenever the salt condition is increasing, their growth is declining. That is non-halophilic organism. Okay. Look for another one. It is halotolerant organism. They will be showing a graph in this manner. This is halotolerant organism. Halotolerant organism, in the sense, if there is salt condition, they won't like salt condition, but even though they will grow, or they will withstand, or they can tolerate the salt condition up to a certain extent. That is halotolerant organism. If you're looking for another one, this is this organism is moderate halophilic. Moderate halophilic. Moderate halophilic. Moderate halophilic means they requires an optimal level of NaCl or optimal level of salt condition they require. More than that particular content, they will die. Lesser than that particular amount of NaCl content also they will die. They require a moderate amount of NaCl condition. Okay. And the last group of graph that is in this manner. This is the next group. This is extreme halophilic. This is extreme halophilic. This group can be called as extreme halophilic. Just look into this diagram. What is happening here? They won't start their growth until they are getting enough amount of NaCl. Whenever they are getting the required amount of NaCl for their growth, then only they are growing, right? So that is extreme halophilic organism. With respect to the increase of NaCl, after that they are growing very easily. This is the diagrammatic representation or the graphical representation of different organism based on their osmotolerance. Okay, whether it is clear?
Yes, sir.
Yes, sir.
Based on temperature. If you are based on temperature, I have already told in previous class, based on temperature, I can classify the organism into psychrophile. Psychrophiles are the psychrophiles. They are requiring low temperature. So they are cold living organism in low temperature. And you can see mesophiles. Mesophiles are present. The mesophiles are present in moderate temperature. They requires moderate temperature. Moderate temperature they requires, pathogens, for example. Pathogens which are causing disease to human beings, not only to human beings, but also to organisms, that is pathogens, they requires moderate temperature. That's why, in human body, the bacteria can easily grow. Our body temperature is 36.9. That is a moderate temperature. In that temperature, this organism will easily grow. That's why when if a pathogen is entering into our body will secrete pyrogens. Pyrogens will increase the body temperature. Body temperature, if the body temperature is increasing, then these organisms cannot live there. And the last group will be thermophilic organism. Thermophiles will be there. And what will be the specialty of thermophiles? They will be requiring high temperature. Thermophiles will be requiring high temperature. In this manner, we can classify. If it is based on pH, if it is based on pH, you can classify the organisms into acidophiles. And you can classify the organisms into alkalophiles. Alkalophiles are there. And you can classify the organisms into neutrophiles. Neutrophiles are there. Do you remember in our blood group there is a neutrophil? In our blood there is a neutrophil, right? In our blood there is eosinophil. In our blood you can see eosinophils are there. In our blood there is basophils are there. Basophil. In our blood there is neutrophil. Isn't it? We are having WBCs in this manner. From where they got their name, From the neutrophil, from where the neutrophil got their name, from where the basophil got their name, from where the eosinophil got their name, Any idea? The neutrophils are getting color during neutral dye. If we are using neutral dyes, the neutrophils will be capture the color if we are using neutral dye. In the same manner, we can color the basophil by using basic dyes. And eosin is the name of an acid dye. Okay, eosin is the name of an acid dye. So by using the acid eosin, you can color that particular WPCs. That's why they got the name eosinophil. It is clear?
Yes, sir.
Okay. So based on pressure also, based on pressure, we can classify the microorganism. The organisms which like pressure is known as barophilic organisms. If they require or if they like pressure, that can be called as barophilic organisms. They can called as barophilic organisms. That is clear. Okay. So based on the concentration of oxygen, okay, based on oxygen concentration, if you are classifying based on the oxygen concentration, O2 concentration, based on oxygen concentration, there will be the first group will be obligate aerobes, isn't it? Obligate aerobes will be there. Obligate aerobes will be there. What is the specialty of obligate aerobe? They strictly, there is no other choice, strictly require oxygen for growth. They strictly requires oxygen for growth. That is obligate aerobes. The second group is known as facultative anaerobes are there. Facultative anaerobes are there. What is facultative anaerobes? You know, they are anaerobes. Anaerobes. There is anaerobes. That is, they are growing in the absence of oxygen, but they grows in high. They are anaerobes, but they are grows high in presence of oxygen. That can be called as facultative anaerobes. The third group can be called as aerotolerant anaerobes. Aerotolerant anaerobes are there. They ignore oxygen. They grow equivalent. They grows equivalent in presence and absence of oxygen. If there is oxygen, they will grow. If there is the oxygen is absent, they will grow. That is aerotolerant anaerobes. The fourth group is known as obligate. The fourth group is known as obligate anaerobes. Obligate anaerobes. What is obligate anaerobes? They can't grow. They can't grow in the presence of oxygen. They can't grow in the presence of oxygen. That is obligate anaerobes. But is facultative anaerobes? They are anaerobes, but they grow highly in the presence of oxygen. But obligate anaerobes, they can't grow in the presence of oxygen. That is obligate anaerobes. The fifth group is microaerophiles. Microaerophiles are there. Microaerophiles are there. They doesn't require normal level of oxygen. They don't want the normal level of oxygen. A little level of oxygen is required. 2 to 10% only. 2 to 10% is required. Only 2 to 10% is required for microaerophiles. In this manner, we can classify the microorganisms. We can classify the microorganism on the basis of the oxygen concentration. The basis of oxygen concentration, we can classify in this manner. Whether it is clear?
Yes, sir.
We are moving forward. Let me take 1 minute break, okay? Just wait here. Just have to drink a cup of water. So the next we are discussing about a few terms. That's about the control of microorganism. For the control of microorganism, we are using different methods, right? Control of microorganisms. First one, what is sterilization? We are using a method that is known as sterilization. Sterilization is a technique. What is sterilization? That is the process by which all living cells or spores and acellular entities from an object or habitat, they are removing or we are killing all the things, isn't it? That is sterilization. So we are killing, we can tell in this manner, killing all living cells, all living cells, all living cells. We are killing all the living cells or spores. We are removing all the spores and cellular entities. Cellular entities. Cellular entities. In the last class, we were discussing about certain cellular entities. For example, prions, virus, like cellular entities are removed from an object. That is the process is known as sterilization. That is sterilization. Okay. So what is disinfection? Disinfection. There is another term, disinfection. What is disinfection? You know, this disinfection, that disinfection that we perform, that is used on inanimate objects. Inanimate objects only. In inanimate objects only, we can use the term disinfection. In disinfection, we'll use chemicals. That we are using chemicals. We are using chemicals. Whereas in sterilization, there is a procedure regarding that we will discuss in later. Anyhow, in disinfection, that is, you can use that term in inanimate objects only, where we are using chemicals. If you are doing disinfection, you know, spores and few microbes, remains. So even if we are doing the disinfection, you know, spores and some microbes will be still there even if we are doing disinfection. Another terminology is sanitation. What is sanitation? Sanitation is related to disinfection itself. That is, microbial populations are removed. Microbial population, not completely, okay? Microbial population are removed, safe to the level of, listen to that point very well, safe to the level of public health standard. Safe to the level of public health standard. Safe to the level of public health standard. That is known as sanitation. So sterilization is there, disinfection is there, and sanitation is there. And another terminology that we are using, that is antiseptics. This antiseptics that we are used on tissues. If you are using on tissues, living things, you can call it as antiseptics. That is chemical agents applied to prevent infection. Chemical agents, we are applying chemical agents to tissues. Chemical agents to prevent infection. If you are using chemical agents to prevent infection, such chemicals can be called as antiseptics. If the terminology side is coming at the last, you know, side, if it is coming, we can tell it as killing. It is used for killing. It will be used for killing. If at the end, if static is coming, at the end static is coming for a chemical, that we use it for stop growth. For stopping growth. For example, if I am drawing a graph, okay. This was a microbial growth, okay. This was a microbial growth. If it is static, you know, if it is static, then the graph will change in this manner. It is static. It is static. If it is side, you know, if it is side, what will happen, you know, if it is side, After this, the growth will decline. That is side. If you are using side chemicals, side in the manner, at the end, if there is side, that will be used for killing. So the growth will decline. And static means they are stopping growth, only they are not killing. So the graph will be moving in this manner. I hope it is clear. Well, that is clear?
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
So we can discuss about some other terms. That is, what is bioremediation? Bioremediation. What is bioremediation? Bioremediation is the process of removal of pollutants. Removal of pollutants. We are removing pollutants. Removal of pollutants using biological agents. Using biological agents. Using biological agents. You can call it as bioremediation. Okay. For example, you might be heard about superbugs. Example, superbugs. Superbugs, they are actually, what is superbugs actually? Pseudomonas, right? Pseudomonas, pseudomonas. Pseudomonas is the superbug. It is used to remove hydrocarbons from oil wells, that is superbugs, bioremediation. Okay. Another term, what is bioaugmentation? Bioaugmentation. What is bioaugmentation? If you are introducing microbial culture, and nutrients, to polluted area, introducing microbial culture and nutrients to polluted area, that is known as bioaugmentation. That can be called as bioaugmentation. What is biostimulation? What is biostimulation? Bacteria will be present in biostimulation. The bacteria will be present in that environment, in that polluted environment. So what we will do, we will promote the growth of that bacteria by adding nutrients. So in biostimulation, bacteria will be present. Bacteria will be present in polluted environment. So what we'll do, we stimulate their growth. How we stimulate their growth? Stimulate their growth by adding nutrients. By the addition of nutrients, we will stimulate their growth. That is known as biostimulation. Okay. I hope the things are clear. Okay. So in this manner, if you are using algae, that is known as phycoremediation. All these things you can expect for your B part. Okay. Phycoremediation. That is phycoremediation. If you are using algae, that can be called as phycoremediation. And mycoremediation. What is mycoremediation? If you are using fungus, that can be called as mycoremediation. If you are using plants, that is known as phytoremediation. Phytoremediation is a process of using plants. If you are using plants, we can call it as phytoremediation. Okay. And you might be heard about the term microbial bioleaching. What is microbial bioleaching? Microbial bioleaching means we are extracting metals from the ores using bacteria. We can extract metals from the ores. That is known as microbial bioleaching. So they will produce by using the sulfur which is coming from the microorganism, which is produced by the microorganism only. They are doing this process. Don't go into details. What is microbial bio leaching? That's only needed. So if we are looking into phytoremediation, if you are looking into the phytoremediation, if you are looking into phytoremediation, in phytoremediation, you can see phyto extraction. The first one is phyto extraction. Phyto extraction is the first thing. So that is use of pollutant accumulating plants. Use of pollutant accumulating. We have to use the pollutant accumulating plants. That is phyto extraction. Then second thing is phytodegradation. 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 In phytodegradation, use of plants and associated microorganisms. Use of plants and associated plants and their associated microbes. Associated microbes to remove pollutant, or chemicals. We are using microbes and the plants and their associated microbes. Rhizofiltration. Use of plant root. We are using plant root to absorb or adsorb the pollutant, from water. Pollutants from water, that is known as rhizofiltration. The fourth one is phytostabilization. Phytostabilization, where we are using the plant, to reduce the bioavailability of the pollutants, in the environment. Okay, that is phytostabilization. Okay, the phytostabilization, that we are using plant to reduce the bioavailability of the pollutants in the environment. Then the last one is phytovolatilization. Okay, use of plants. Here we are using plants to volatilize the pollutants. We are using the plants. That is phytovolatilization. And the common terms that you may be knowing very well, what is bioaccumulation. So we are discussing about several terms. So each and every words, you should know each and every terms. What is that? You can expect question from all those regions. Bioaccumulation. Accumulation of, what is accumulation? That is very clear. Accumulation of a chemical. That should be toxic. Okay, that is a toxic chemical in their body. Accumulation of a chemical in their body during the lifetime. Such chemicals cannot be metabolized. Cannot metabolize or excrete. So that will be present in that organism's body. Such process is known as bioaccumulation. What is biomagnification? Biomagnification. What is biomagnification? Biomagnification means, so this bioaccumulation. Due to the bioaccumulation, continuous bioaccumulation causes the increase in concentration of this chemical in organism's body, and they will be passed on to the succeeding generation. So, bioaccumulation. Biomagnification is the result of bioaccumulation. Bioaccumulation leads to the increase. Bioaccumulation can lead to the increase of the chemical, in tissues, successively higher in trophic levels. So higher trophic level, if you are going to higher and trophic level, you can see the increase of that chemical. For example, in the case of microbes, the amount of that chemical will be very less. Or if you're looking into the fishes, small fishes, it will be some little bit higher. When it is going into higher fishes, and it is coming into mammals. Or simplest example is DDT. When DDT is present in embryos also, so that's in that much is the extent of accumulation of these chemicals. Okay, that is clear. So what is phytoremediation? What is phytoremediation? What is phytoremediation? That is remediation. You know what is bioremediation? That we have already discussed about the bioremediation. In phytoremediation, what we are using, is done with the help of phytoremediation is done with the help of phytochelatins. That is known as that is known as phytoremediation. That can be called as phytoremediation. So, glutathione is a chemical. Glutathione. That is GLU 16 and glycine. This is glutathione. When two glutathiones joins together, glycine plus glycine. What will happen, they will remove one molecule of glycine. They will eliminate one molecule of glycine, and you will get cysteine and glycine. This compound, which is having this compound is known as phytogelatin. This compound is known as phytogelatin. So in cysteine, if you are looking into cysteine, very well, we have discussed about in amino acids, cysteine and methionines were sulfur containing amino acids. So sulfur present in cysteine. Cysteine is having sulfur. The sulfur present in cysteine is responsible for scavenging. Scavenging the metal. And store the chemical in vacuum. That is known as and that remediation is known as phytoremediation. Okay. Then for bioleaching, which type of organism? Bioleaching, we have already told about bioleaching. Which type of organism? Microbial bioleaching we have told. Some examples for bioleaching. Just note out few examples for bioleaching. We can use leptospirillum. Leptospirillum. You can use leptospirillum. Thiobacillus. You can use Thiobacillus. Thiobacillus thioxidants. All these organisms we can use. You can use for bioleach. Okay, that's clear. So another organism, this is another special organism, that is Deinococcus. Deinococcus radiodurans. Deinococcus radiodurans. What is the specialty of this organism? You know, they can withstand high radiation. This is a microorganism that can withstand high radiation. They will withstand high radiation. So it can be used to extract. It can be used to extract radiation emitting compounds. Used to extract radiation emitting compounds. Radiation emitting compounds can be extracted with the help of Deinococcus radiodurans. That's also clear. So in microbes, we have to study about few diseases. Okay, that also we can discuss in today's session, and we can wind up. Okay, so plague. Plague is a disease. It is caused by Yersinia pestis. Yersinia pestis is causing plague. Typhoid, you may be knowing very well. Typhus fever. Typhus fever is caused by rickettsia. Rickettsia. Rickettsia browsing. Q fever is caused by Oxyella brunette. Scarlet fever is caused by Streptococcus purulens. Lyme disease that is caused by Borrelia. Another one, ulcer. Ulcer is caused by Helicobacter pylori. Another disease is caused by Legionella pneumophilia. Okay. So these are some of the diseases caused by microorganisms. Not over. Another disease, that is toxic shock. This disease is caused by Staphylococcus aureus. Another disease, meningitis. It is caused by Neisseria meningitis. Another disease, pneumonia. Know very well, that is Haemophilus influenza, right? Haemophilus Influenza, Haemophilus influenza. Some foodborne diseases. Foodborne can be transmitted through food poisoning, botulism. This is caused by Clostridium botulinum. Clostridium botulinum, right? Clostridium botulinum. Cholera. Cholera is caused by Vibrio cholerae. Vibrio cholerae. Shigella. Shigella is caused by shigellosis. Traveler's diarrhea. Traveler's diarrhea. Caused by E. coli. Now some of the bacterial diseases are over, foodborne diseases are over, and we can look into some of the protozoan diseases. Protozoan disease. Toxoplasmosis. Toxoplasmosis caused by toxoplasma. African sleeping sickness. African sleeping sickness. African sleeping sickness that is caused by trypanosoma. Another one, leishmaniosis. Leishmaniosis is caused by leishmaniasis. Malaria that is caused by Plasmodium falciparum. Three types of organisms are there: Plasmodium malaria, Plasmodium vivax, and Plasmodium falciparum. Okay. And if you're looking for some viral diseases caused by virus, rabies, you know very well about rabies. Smallpox disease caused by virus. Influenza is causing several diseases. Prion disease. One example for prion disease that you have to study. Prion. Scuru. The same manner. Palata. Familial. Insomnia. Okay. So these are the some of the diseases which is caused by the microorganism, pathogenic microorganisms. Okay. So with this, we can wind up today's session. So in today's classes, we were exclusively discussing about microorganism, their growth curve, and based on their growth curve, we have discussed about different equations we have made from that. And at the last session, we were discussing about some general topic. For that, from the first session, you will be getting question for C part, and from the second half, you will be getting question for your B part also. Let me wind up the recording. Just a second.